at Harborview. She's the director of the Harborview Senior Care Clinic. She leads the MSTAR program here at UW and has won mentoring awards for that multiple times. But in her humility, you may not know that she's an internationally known researcher also for her work, focusing particularly on evaluating the effects of the extracellular matrix on tissue repair and microvascular aging, and a lot beyond that. She's also done a ton of work focused on frailty and working with the surgeons here at Harborview and leading our Jerry Clinical Council. So May wears many hats and wears them well and with humility. And I'm excited to hear about the Dog Aging Project and the work that you're leading with that.
Okay, thank you, everybody, and thanks, Kate, for that kind introduction. I want to start out with two caveats. One is that much of what I'm going to say today is my opinion about where geroscience is heading for humans. There is lots of ways of looking through the lens to see where this is all going to land for aging, health span, as well as lifespan, maybe. And that these slides largely belong to the Dog Aging Project, and the PIs of the Dog Aging Project, an amazing group that's really started this back in, well, it's 2018 officially, but probably was in the eyes of the beholders, which was Matt Kaverlin, Katie Creevy, and Daniel Promislow several years before that. So those are my two caveats. I have no disclosures. The objectives today are to review the background of geroscience and its ongoing evolution, updates on the Dog Aging Project, update you on triad and rapamycin as a geroscience intervention, and then looking ahead in a rapidly changing field. So this is the classic slide about the pillars of aging, and we have to show this at the beginning of every talk on biology of aging and geroscience, because these pillars underscore the fact that if you attack a specific disease, you may delay that disease, heart disease, cancer, etc., but you won't impact the overall aging progression that a human or any organism undergoes. And the concept of the pillars is that if you instead look at specific programmatic changes that occur in most cells or all cells, etc., you may impact multiple diseases, cancer, heart disease, dementia. And so as this field has evolved, it has shown that there are more and more pillars that we might go after in order to impact both health span and maybe even lifespan. And I remember when I was starting in this field, the big thing was telomere attrition. That was the thing that was going to improve all of the aging paradigms. And of course, that's just one of many features of aging cells. And so now we have many more pillars of aging, including, the latest might be microbiome, in your opinion, or some other epigenetics, if that's your area of interest. But the concept here in biology of aging is to take these pillars of aging and make a translational impact, which is now what we call geroscience. So geroscience is sort of the translational impact of biology of aging. So just to remind you that one could argue about when this started, but I think most people in the field that believe that the concept that started much of this and the development of those many pillars of aging was caloric restriction. And as many of you might recall, caloric restriction was, I think, highlighted at least in a major way by this landmark study in rhesus monkeys that was published in Science in 2009. And to make a long story short, they were 27-year-old matched rhesus monkeys. They were not necessarily litter mates, of course, because rhesus monkeys don't have huge litters, but there was an intervention of caloric restriction started around young to middle age. And there were these marked changes, including improvements in less cancer, less cardiovascular disease, less diabetes, less brain atrophy. And this really launched what I think was the field of geroscience, although there were experiments prior to that indicating that restricting your caloric intake might make an impact. But this was probably the pivotal study that underscored that we need to look at what pathways then were impacted by caloric restriction to better understand biology of aging and to create the beginning of those pillars of aging I alluded to. Now, where did that land in terms of human data? Again, this is my perspective of sort of what I think were the pivotal studies, but there were many, many more. The big one in human data, in my opinion, is the calorie study. It was based out of Duke. They tried to get several hundred humans to caloric restrict, this time 20% for 2 years. They weren't quite able to do 20%. They landed at around 12%. But this group of adults who did this for 2 years, and again, what's interesting here, if I didn't note this already, is that it was started at middle age in this case in humans, and the caloric restriction was all intake. It wasn't just, carbs or fats or anything like that. And this group is continuing to highlight that caloric restriction, even if it's done for a 2 year period of time in middle age, still has some changes that might be not only useful for health span, but might be useful for lifespan and other features of the aging process. So what else works then? Well, the big thing now, as I said, caloric restriction works, but people said, well, caloric restriction is really hard for humans. The calorie study certainly showed that. So what has evolved, as you probably know, is the whole idea of intermittent and periodic fasting. The idea here is, well, instead of just, suffering with caloric restriction, and we know that humans could probably do 12%, let's talk about just intermittently caloric restriction. And those of you who followed this field know that there was this 5 days off, 2 days on, 1 day off, 2 days. And there are all kinds of combinations that might work for some people, might not work for others, and the field got very, very muddied. But I think even if you said, well, they're just eating less because they have less time to eat, there was this idea that something here is beneficial to the pillars of aging and therefore delaying some of the diseases of aging. And where is this field heading? As you probably know, is most people think that intermittent fasting is difficult for adults. There was this concern that it might be even dangerous, but that really hasn't panned out. In other words, if you suddenly take in a huge amount of calories that you after a period of caloric restriction, maybe that's dangerous for cardiovascular events. That hasn't really panned out. But there was this idea, let's make this simple. Maybe we just need to do timed restricted eating. That is to say, instead of intermittent fasting for a day in a week, or 2 days in 2 weeks, or 1 day in 5 days, how about just eating during a restricted period of time each day? Where is this field headed? Again, lots of caveats here, lots of individualized variation, lots of muddying the waters when you study people who already have metabolic syndrome, fatty liver disease, diabetes type 2. But there is a signal in a sense that eating during a restricted period of time every day, which might be easier for a typical person, has benefits on metabolic parameters, many of the pillars of aging, and again, potential for mitigating age-related diseases and improving health span. One of the things that people are considering is that, okay, how much do you have to restrict your time? The sense is that it's impossible to do less than 8 hours. That is to say, eating for most adults, that is to say, you need at least 8 hours where you're allowed to eat. Probably if you go beyond 10 hours, you lose the benefit of time restricted eating. And now there's also a lot of interest in saying if you're going to do time restricted eating, you really should do it in a way that matches your diurnal rhythm. That is to say, you probably should have your on time, which is when you start eating, about an hour or two after you wake up. So have your coffee, but don't have it with a ton of cream. And then an hour or two later, go ahead and start your eating period. And then very much recommended that you stop the eating a few hours before you go to bed. And if at all possible, do go to bed in a time that matches circadian rhythms and light-dark cycles. So I think this is where we're at now. Again, a lot of this is my opinion. People will say, well, I saw this study that it was just that people eat less because they don't have time to eat more. And actually, that's probably not the case. But there are a lot of caveats to this. And as I said at the beginning, this is my sense of where this field is heading. Okay, so we know that the typical person, and certainly typical people in the United States, modifying diet is hard. And even if you say it's going to be time restricted eating and not necessarily something more difficult like intermittent fasting or even more difficult like chronic caloric restriction, what about just slowing aging and improving some of those pillars of aging with a pill? And as many of you know, there was a lot of interest a long time ago, and maybe still to this day. There's multiple clinical trials of senolytics. So, what are senolytics? Well, the concept is as you get older, you have more and more cells that are not proliferative, but they're not dying either. They're just sort of sitting there, releasing oxidative stress, creating havoc with inflammation, cytokines, et cetera, and that you need to get rid of those cells. And of course, this is a field that was started and largely advanced by Judy Campisi, who unfortunately passed several years ago. But this idea of senescence-associated secretory proteins are bad, and we need to get rid of these cells. And so that's where the field of senolytics started several decades ago and continues to this day. And the caveat always is, all right, if you get rid of senolytic cells by inducing apoptosis, but you're 90 years old, you have stem cells to replace those cells that are now gone, whether it's in the skin or in other tissues. And of course, there's always a question, some of these agents cross the blood-brain barrier to get into the brain to get rid of the senescent cells in the brain? So a lot of interest in this field, but I think it's being supplanted by some of the other things we'll talk about, which is slowing aging with pills that target nutrient signaling. So again, as I told you, a lot of the pillars of aging have been developed based on that original paradigm of caloric restriction. So it wasn't surprising that signaling pathways that were impacted by caloric restriction, intermittent fasting, time restricted eating were also a target. And here's where we have a lot of pills that are in study today. I mean, we're talking about dozens of clinical trials across the United States. And of course, we'll talk a little bit more about rapamycin, but we know there's metformin trials going. Our own Angela Hansen is involved in a metformin trial. Don't ask me about resveratrol. We know that alcohol is bad. Even 1 to 2 glasses of red wine a day is probably not good for you. That's the latest data. So resveratrol, which is the active ingredient in red wine, is really not being very well studied anymore for good reason. Melatonin is in here. I laugh about curcumin or turmeric. That's in many of these pathways, just to indicate that nutrient signaling is probably of greater interest right now than senolytics. Where is this field heading? Well, not surprising. GLP-1 receptor agonists and SGL2 inhibitors are now added to the list. Our own Dr. Ray gave you a presentation on her work with SGL2 inhibitors and GLP-1 agonists. Well, yeah, Elizabeth could correct me on whether she's studying both groups or limiting to one right now. But these are very much under study because, as I indicated, people like taking a pill, and they're increasing data indicating that GLP-1 receptor agonists are impactful in the pillars of aging. And again, the improvement in health span. And of course, SGL2 inhibitors are also along that pathway. And also, this has a recent study saying that they act as a senolytic and therefore can have multiple pathways by which they will impact the pillars of aging. So where are we now? Again, my opinion on anti-aging interventions. One could say, May, you didn't mention this or you didn't mention that, but I think that this is a good summary, and I love the little diagram of some of the drugs that I already talked to you about, the senolytics, the the anti-diabetic drugs, metformin, SGL2 inhibitors, GLP-1 receptor agonists. And I think that rapalogs, of course, which we will talk about shortly, which are involved as mTOR inhibitors, but also involved in many of the pillars of aging. And then I think, in my opinion, the one group of interventions that is going to get increasing press is going to be the metabolic interventions based on NAD pathway. The NAD pathway, for those of you who remember it from medical school or grad school, is extremely complicated. Part of how you get into the NAD pathway is by dietary um, intake with compounds such as those that contain tryptophan, but there's also scavenger pathways, so that once you take in dietary interventions, they interact with endogenous pathways within your cells, especially muscle cells and other metabolically active cells, to create these compounds that, when properly targeted, again, here we go again, impact those pillars of aging. And I have a lot of colleagues who feel that NAD supplements are going to be the next big and the next big thing in anti-aging research, just because when taken properly, they have a very low side effect profile. And for the interest of the American consumer, they do not require a prescription because a lot of them are available online. So stay tuned for this class of mediators. So looking back then, I wanted to just show this overall diagram of where I think the timeline has come over it's going to be almost a century when you talk about, I won't talk about the Minnesota experiment, but that was one of the early quote unquote experiments on humans that were caloric restricted. Talked about caloric restriction. Along the way, I did not talk about Biosphere 2. Many of you weren't even born then, but that was an accidental intervention for caloric restriction when scientists entered a bubble and were supposed to feed themselves for 2 years, and then they couldn't, so they were caloric restricted. Those folks are still being studied to this day. Talked about calorie, intermittent fasting, and time restricted eating. And then on the top line, some of the interventions that have come that are therapeutic interventions, many of which are available in oral form. And just for my colleagues who remember some of the classic studies, I put them down here just because they don't get enough press. A lot of times people forget to highlight the giants in the field that contributed to this. Roy Walford with caloric restriction back in 1992. Rudman study in NEJM 1990, for those of you who remember, about the somatomedin path hypothesis of growth hormone and IGF-1, which turned out to be not very practical, but has led to its own field of people who still believe in the GHRH growth hormone pathway. And then Rickman's studies that have been many years ago and often do not get enough highlights in recent literature reviews. Okay, so where else are we heading? I wanted to allude to the CTIP program. Why? Because this is where a lot of the new experimental interventions are going to launch. It was a program that's been available since 2013 at NIH. It is still it's still going on to this day, and I know because I just got appointed to the CTIP program. I have no idea what I'm going to be asked to do, but best that I can tell, it offers the research community the opportunity to submit compounds that you might want tested to see if they can impact the pillars of aging. The testing is then done at 3 sites: Rutgers, the Buck Institute, and the University of Oregon. There's the opportunity for in-house testing as well in the NIH campus for compounds that come through via suggestions. To the broader community, there's a nomination program which is interviewed by the Access Panel, which then recommends to the CITP steering committee whether a compound should be tested. I have been appointed to the Access Panel, and it's going to be very interesting because this affords people to sort of think outside of the box of new concepts that might not have been considered previously, whether it's a repurposing of an existing drug or trying an old compound that might not have been considered by the usual scientific analysis. Okay, so we think that worms are already and will increasingly be used to deploy not only the idea of new compounds, but whether or not these compounds will work for, say, you. And the reason that that can be done is that worms, as many of you know, C. elegans and specifically, can be modified to have certain genetic traits that may or may not, depending on your perspective, reflect a human. And so compounds can be rapidly tested and assayed for outcome measures, whether it's lifespan, or with worms, it's often their ability to move well or have phenotypic evidence of being healthy. And because they can be genetically modified to reflect specific genetic diseases that you or someone might have, this field is going to rapidly progress to not only be able to do a high throughput testing of compounds, but also to be able to do that in the context of reflecting an individualized genetic risk. Okay, so that brings us to, so that's my perspective on geroscience and where it's headed. Now we're going to switch to the dog aging project. And the reason to do that is because dogs, as I've alluded to before, have a compressed model of aging and share many of the features of aging in humans, but also share our environment. And that is true for those of you who have dogs, and for those of you who've witnessed some of the dogs that your friends or your loved ones or your family have, that you know they are very much a part of our environment. They are very much a part of our family. People keep track of their dogs in a way that they don't even keep track of other members of their family. And they are genetically diverse. And unfortunately, they age very rapidly. So many of us lose our dogs long before we feel that we would have aged, because as you might know, the first year of a dog is 15 years, the second year of a dog is 9 human years, and then it's the classic 7-to-1 ratio. So they have a very rapid age progression, and that rapid age progression is accelerated by body size, which is another feature that has been fairly consistent over time, that larger dogs have a shorter lifespan than smaller dogs. And I want to say many of these slides are from the Dog Aging Project. I provided the references as I was able to find them, but we can, if anyone wants primary references for any of these figures, the Dog Aging Project and the consortium keeps an amazing record of the dozens and dozens of publications that have come out of this amazing project. So where is the DAP right now? It's over 50,000 dogs. They're still being added to, but largely the idea now is we have enough dogs to that are not only middle age, but also approaching advanced age, that much of the focus will be on looking at mechanisms, interactions, and translatability to the human condition. So the data is publicly available. There are thousands of data points on these dogs, starting with the health survey that the owner does, which has almost 5,000 questions, and then the consistent follow-up assessment provided by owners, as well as the collection of samples and other more detailed testing on a subset of the dogs. This data is all publicly available, and I might have lost track of how many publications, again, that this amazing project has generated. So I already talked about how dogs have compressed aging because of their accelerated organismal aging. That's, again, worsened by size. The other thing that we know, though, that the heterogeneity in size is actually not only, again, helps us understand why do older dogs have the shortened lifespan, but it provides a feature of what should I say, uniqueness among the dogs that also might allow us to study some of the aspects of the heterogeneity of aging and human size. So, you know, we all know about this is Kamal Chada's 2 dogs. One is 8 pounds, one is 110 pounds. And I'm always worried that the 110-pound one is going to eat the 8-pound one, but they seem to get along great. But we think that this heterogeneity in size is a strength rather than a concern about the Dog Aging Project and its data collection. So the heterogeneity in genetics is also terrific. meaning you couldn't have asked for better distribution. This is old data points again, because there's over 50,000 dogs, but holds true to this day, which is that it's a 50-50 mix, nearly perfect, of purebred and mixed bred dogs. So we've got the purebreds, and then we've got the dogs. We're not quite sure where the combination is, but definitely mixed breed. So somebody always asks, what about the fact that most dogs in the United States are sterilized, spayed, neutered, whatever term you want to use? And that is the case. The majority of these dogs, because they're companion dogs, are in fact spayed or neutered, but there's enough data points of intact males and intact females to have additional information regarding sex as a variable and in the non-neutered state. So how are dogs and humans similar then? Let's think about that. Both exhibit cognitive decline with aging. Cardiac hypertrophy is seen very commonly in dogs. And again, for humans, as you know, for humans that don't have underlying severe coronary artery disease or an underlying cardiomyopathy, hypertrophy of the myocardium leads to HFpEF, heart failure due to preserved ejection fraction, as the dominant cause of heart failure in older adults. Dogs don't get it as severely as humans, but they can exhibit systolic hypertension. They don't get the increase in pulse pressure, but they do have a slow but steady increase in systolic blood pressure in a subset of dogs. Kidney disease is present, sarcopenia and frailty. We're not sure about osteopenia because that would require DEXA scanning. They do have prostate changes. In fact, beagles are a model of benign prostatic hypertrophy as well as prostate cancer, and they do have changes in the immune system. How are they different? I already talked about lifespan has less size dependence. They do not get atherosclerosis. They tend to have normal diastolic blood pressure. The cancers that they get tend to be lymphoma and sarcoma, which I know many of you, including me, have experienced personally in our pets. Trauma is a major cause of death, but it's not ground level falls. It's running out and chasing cars. And they have different environmental risks because of the way they're fed. But let's talk about some of our favorite topics in aging research, and how to mitigate would be what is going on with cognitive decline in dogs, and how can that be studied as a way to mitigate the features and the impact, the risk factors for cognitive decline in humans. The cognitive decline in dogs has disorientation, social withdrawal, sleep disturbance, incontinence, and decreased activity. Dogs do get A beta, classic plaques in their brain. They do not get classic tangles. And this has been an area of ongoing research. So the A beta staining and the A beta prevalence with what we call canine cognitive dysfunction is very similar, not only in regional distribution, but in morphology with humans. Tau, on the other hand, is not as well studied. Part of the problem is that as of right now, last time I checked, we could not detect tau in the CSF or blood of dogs, although the sample size is very small. But many people have reported finding tau in histologic sections of dog brains who are older or who have canine cognitive dysfunction, but it's not been well clarified which forms of tau are found and whether the morphology and the distribution is similar to humans. How do we test for cognitive dysfunction in dogs? Our colleague in the DAP, Evan McLean, has developed some beautiful measures of cognitive dysfunction in dogs. So in addition to those subjective measures that I showed you on the previous slide that are reported by owners, there are actually testing features that dogs in the Dog Aging Project undergo each year, including, as I said, the owner-reported survey, but there's additional specific tests of cognitive function, including 1, 2, 3 treat, and treat, hide, and seek. These are very hard to do, and uptake by owners is it takes very motivated owners to report these, but nonetheless, Evan and his colleagues have had enough success in getting these reports and tying them into other features of the dog's cognitive domains to understand which tests work, how their predictive value, and what that means for aging in the dog. One of the observations is, of course, it's related to age, just like it is in humans. But interesting enough, it doesn't appear that big dogs have a faster cognitive decline um, um, as they um, as they do for other features of aging. That is to say, big dogs have a higher incidence of cancer and have a shortened lifespan, but they do not appear to have a higher risk for cognitive decline when they are matched against smaller dogs in aged matching. So weight is not a driver for cognitive decline as it is for other age-related diseases in dogs. But talking about age cancers in dogs, um, I want to highlight that that's also becoming more and more controversial with respect to the size relationship. When we talk about cognition, though, we have to talk about frailty, because frailty is again another huge component of health span for older adults. If you can avoid frailty, which is now increasingly called resilience, although we could argue about how to define resilience, it's not exactly the opposite of frailty, but it is a increasing area of research in gerontology. We very much wanted to have a similar frailty paradigm for dogs, and that has also been developed through the Dog Aging Project. So human frailty, as you know from the Freed criteria, is weakness, weight loss, exhaustion, slowness, physical activity. And sure enough, the Dog Aging Project colleagues led by Katie Creevy and many of the other investigators have developed a dog frailty index, which has been called FIDO, and it has many of the features of the Freed frailty index, that is physical condition, physical activity, mobility, and strength. Again, much of this has to be done by the very motivated owners that have dogs in the Dog Aging Project and their testing. But this study has, in many, many cases, shown that owner-reported activity measures and strength measures are highly correlated with objective data. That is to say, for example, that a subset of dogs in the Dog Aging Project have actigraphy monitors, and which are very objective ways to measure dog mobility and activity. And it has been shown that the owner report correlates very highly with the data that's been collected on the subset of dogs that have actigraphy monitors. So so one of the things that we're very interested in then, of course, within the frailty paradigm is walking speed. And that has also been looked at, as I showed you, along with mobility and going up and down stairs, etc. It's interesting to note that walking speed in dogs drops off around age 7, kind and fairly independent of dog size. And if you do the math, as I alluded to earlier, of first year is 15 years, second year is 9 years, and then 7 to 1, that correlates to about 60 years old. I hope that's not the case for me. I plan to run around and maintain my walking speed well beyond my 60s, but this is just to highlight that dogs do have that drop off at a time that approximates when we would consider a human to be quote unquote older, although I think most of us appreciate that that should be moved off into the 70s or 80s now. So I alluded to earlier, then we were talking about cognition, we're talking about frailty. What are the shared exposures with humans that also makes this data so compelling? This is data from the Dog Aging Project again, Audrey Ruppel and her colleagues, published in journals like Science. that shows you just a small fraction of the environment that we share with dogs that allows us to then look at these exposome variables and how they may impact the development of impaired cognition, frailty in a way that you can't do with humans, again, because of that compressed lifespan of our dogs. Things that we think are going to be specifically interesting in the future as more and more of the dog aging project dogs are in middle and advanced age now because they've been in the study for, some of them for the full 7 years, are things such as life space, including stairs, which are documented by their human owner, including how often they use those stairs. Green space, which would be considered both natural green space, including do you live on a farm versus artificial green space? Like my son takes his dog they live in Brooklyn, to a dog park at least once a day, if not twice a day. So those variables are all measured in the exposome research that is being very carefully collected by the Dog Aging Project. What is the else is in there is meal frequency. Why is that important? Well, I alluded to earlier about caloric restriction and then intermittent fasting, and now the interest in time-restricted eating. Is there a signal here in dogs? Well, it appears that there might be a signal here, and dogs provide a unique opportunity to again understand this relationship because of all the features I already told you several times. Now, one could say, well, I have to feed my dog 2 or 3 times a day because it's getting frail. And the once a day fed dog might be a more robust dog. But nonetheless, there is a signal that indicates that time-restricted eating in dogs might in fact be providing some clues as to how time-restricted eating might improve the human condition. Okay, so now we're going to talk a little bit about some of the differences that may both be a challenge when you're studying dogs as well as a benefit. Again, we'll go back to the fact that humans, although we spend a lot of time talking about BMI, and we certainly appreciate that humans that have gigantism or growth hormone excess have excess mortality and a shortened lifespan, there is not that classic correlation between size and age at death in humans like there are in dogs. And we appreciate that, but we feel that that can also be leveraged as a strength. There's also a sense that the distribution of diseases in dogs has some very much similarities, including, as I told you before, cardiac hypertrophy, some cancers, but not many, but not others. We talked about dogs getting CKD, but I also alluded to the fact that dogs have trauma as a major diagnosis. And in terms of impacts upon their well-being and their health span as well as their lifespan. The trauma in dogs is different. As I told you, it's, they got hit by a car because they were chasing a car. So it's not the ground level falls that we see in human type trauma, which is now, as many of you know, the #1 cause of admissions to the trauma ICU at Harborview in older adults is ground level falls. So that is definitely a difference that we want to recognize. And in fact, that always reminds me of the classic compression of morbidity curve, where Fries many years ago in the 1970s, and he published this classic figure in 1981, where he said that the ideal would be to just live, and then boom, get hit by a car, or have trauma as a way to compress morbidity. No one thinks that's a good idea, and we certainly don't want humans to run around chasing cars or falling downstairs or having even ground level falls. But it is sort of this interesting thing about a subset of dogs having that trauma as a sort of a compression of morbidity, where they don't even get to older age because they're very active and they're running out and about and get into trouble in that way. So what are the other differences we should just recognize? I already told you dogs do not get atherosclerosis, but they do get cardiac hypertrophy. They get myocardial deposition of type 1 collagen, which causes fibrosis. So the heart failure signal in dogs and the heart hypertrophy signal in dogs will be useful in the human translation. I already talked to you also about dogs do have a slight increase in a subset of dogs of systolic blood pressure, but they do not have the classic age-related decrease in diastolic blood pressure, which results in an increase in pulse pressure, as we all know in aging, in human aging. That increase in pulse pressure is not seen in dogs. So they do have the systolic changes in a subset, but they're milder and they don't have the diastolic decrease in advanced age that we all recognize. Um, the cancer, I think it bears a little bit of time to review. We already talked about size and cancer diagnosis in older dogs, with dogs that are larger having a higher prevalence of cancer diagnosis. However, it turns out a recent study by in Europe has said that there are many other variables that makes correlation difficult, including heart failure as a mitigating factor. So bigger dogs die earlier of heart failure. So you can't really know all the cancers they might develop. There's the sense that at least in Europe, inbreeding is of larger dogs is a variable in cancer diagnosis. And that although size and proliferative potential, especially for osteosarcoma, is certainly an observation. There are a subset of breeds and driver mutations that make it very difficult to know for sure that a larger dog is truly increased risk of cancer because of their size versus other variables. Again, evolution, inbreeding. And the other thing, the reason why it's important to cite the European data is that Europeans don't fix their dogs. So their dogs are, in fact, in some villages, it's considered like you just take care of your dog so that it doesn't get so it doesn't get pregnant or it doesn't get other, if you have a male dog, so it doesn't get other dogs pregnant. But the 90 percent of neutering or spaying your dogs is much more prevalent in the United States than in Europe. So the Europeans would say some of our data needs, especially on cancer and some of the other features, needs to be viewed in that regard. Okay, so, but nonetheless, the types of cancers that we get relative to dogs are different. Dogs, the list on the dogs is on your left: lymphoma, oral and nasal cancers; humans, as you know, lung, colorectal, breast, and increasingly, unfortunately, pancreatic. All right, so now I just want to show this slide again to talk to you about the anti-aging interventions, because we're now going to say, how did the Dog Aging Project lead to TRIAD? TRIAD is the test of rapamycin in aging dogs. The goal, as many of you know, is whether giving a dog rapamycin can increase health span and lifespan. We all know that rapamycin is probably the most effective longevity drug in laboratory animals, and you saw this slide before, so I will just briefly allude to the fact it was discovered on Easter Island. It is the basis for drug-eluting stents and for many other present-day uses in clinic in humans. And rapalogs are used in, like I said, many domains in terms of immunosuppression, but at the right doses, it appears to modify aging in a way that mimics caloric restriction via mTOR inhibition. Again, this idea of using those pillars of aging that were highlighted by caloric restriction to better understand the aging process. There is a lot of data on how mechanistically mTOR inhibition could slow cell proliferation, induce cell cycle arrest, and mitigate pathways that lead us to age faster. So mTOR inhibition works, and we're very excited about how its mechanisms both where they impact autophagy, protein synthesis, or mitochondrial function, might in fact lead to a signal that promotes health span. So, so these are all well known, and so I don't want to belabor them too much, but the background for triad should be reminded once again because these people did the seminal work on this, and that was work done by folks that treated mice starting at 20 weeks of 20 months of age. So 20 months of age for a mouse is, that's like a 60 to 70-year-old human. And this landmark study showed that treating mice for 12 weeks, beginning at what was late middle age, and if not even late age, improved lifespan. This was published back almost 10 years ago. Then there's been studies including transient rapamycin, again, started at middle age, improving oral health. Transient rapamycin improving cardiac function when started at 20 months of age, which then led to a pilot study in dogs in the Dog Aging Project, where they gave transient rapamycin and got a signal that indicated that there might be improvement in cardiac function in dogs treated for just 10 weeks, again, started in middle age. So that was the backdrop, which I alluded to in a talk, I think two years ago, when Triad was part of the Dog Aging Project. As you know, it is a large randomized trial. I call it clinical trial, even though it's not humans. The dogs have to be healthy and at least 7 years of age and at least 20 kilos. They get treated for 1 year with oral rapamycin once a week with 2 years of follow-up. Lifespan is the primary endpoint, but we have very strong endpoints for heart function, cognitive function, and disease incidence, as well as very careful studies of functional status. The current triad sites are here. There's several more that's been added, including, yay, as of last month, thanks to Shelby Knowles and the Triad team, which I want to recognize this major group of wonderful staff, Anna Ferguson and Shelby Knowles here, but many other wonderful staff based at TAMU and other sites. Many of them are telemed, not telemed, but tele-research sites across the country. We have added yet another site to the Seattle area, including downtown Seattle. So this list of current sites is growing. The reason why you have to have a site is for a dog to be enrolled in Triad. They have to be able to get to a center in order to pick up their medicines and to have either neurologic or cardiac outcomes testing, in addition to all those other measures that I talked about as primary and secondary outcome measures. So Triad is ongoing, and we now have an R01 for Triad and are very enthusiastic. We're in year 1 right now of a 5-year R01. So while that is going, though, people are saying, well, we should probably keep giving rapamycin to humans. And as many of you have probably heard, there was an off-label rapamycin study published back in 2023 now. This was a study published by Matt Caberlin's team, 333 adults. I think many of them were in the Seattle area, were just taking rapamycin. They had a dose that was into the 5 to 10 milligram weekly range. And as you might have remembered, that because the study's now over 2 years old, for those of you who are familiar with it, there was a lot of subjective sense that I feel better, my health has improved, I'm happier, my brain works better, I feel younger, and I have more energy. So that was an observational off-label use of people taking rapamycin for more than 60 days, again, with that median dose in the 5 to 10 milligrams per day, per week oral range. That was the observational study. And now, and I should add, there was only mouth ulceration, canker sores were the only signal that there may be something concerning about taking rapamycin, but there was no other concerns regarding safety. And the median duration of use was well over a couple of months. So what got published recently? Well, not surprisingly, there was an RCT that is obviously it's been ongoing, but it was published a couple of months ago, and that's the Pearl trial. And this was done by a company called Ageless RX. I love it. Ageless RX, based out of Michigan. The Dog Aging Project investigators knew of this study, but not necessarily of these specific investigators. What did they do? Well, this paper, and to the extent that there was time since we just came out, well, we just found out about its publication recently, had a lot of details that I, as a geriatrician, would have liked to have known. Sorry, this is not. There it is. It's decentralized, single center, double blind, placebo controlled, 50 to 85 years. People were selected based on contacting Ageless RX and indicating that they were interested in participating in the study. The primary endpoint was changes in visceral fat as measured by DEXA. Secondary endpoints were changes in lean muscle mass, bone density by DEXA, bunch of blood test results, and then self-reported surveys of quality of life with the SF-36, and then frailty, but then they cited the WOMAC. As many of you know, the WOMAC is a pain scale that's used for arthritis. So I'm not sure whether that was a typo, but the methods were, again, anybody that wanted to participate and contacted Ageless RX via their online medical platform. There were 50, and the range was 50 to 85 years of age. So what could I glean from this paper? They did not indicate a mean age. They did not indicate an age distribution. They did not indicate a sex distribution. They just said there were males and females. 40 got 5 milligrams, 35 got 10 milligrams, 39 got placebo, 48 weeks treatment. The results I'll highlight here with the caveats that, again, I'm not sure who these people were, except that they were between 50 and 85 years of age. They were men and women who contacted Ageless RX via the website. And but what they're reporting was that women who took 10 milligrams of rapamycin had improved lean tissue mass and less self-reported pain using the WOMAC score. So I think they conflated frailty with the WOMAC score. They also had better emotional well-being as measured by the SF-36. And but that was only true for the women taking 5 milligrams of rapamycin. The men showed no benefit in any of the parameters, but again, they didn't make it clear about the age distribution of the women. They're presumably all postmenopausal because you don't want to give rapamycin to someone of childbearing age, but 50, you never know. And again, wasn't clear what the mean age was or what the number of men were relative to women. But no other significant effects were observed. And so the safety signal was very high, which helps us recruit for triad. And, basically this study indicates that more of these studies are going to be coming forth. So numerous studies are already underway. Periodontal disease here by Dr. An, one of our collaborators in the DAP, premature ovarian failure, muscle function and mass coming out of the UK, and then rapamycin and MCI and early Alzheimer's disease coming out of our colleagues at UT San Antonio. So more to come, and I'm happy to bring you up to date a year or two from now. Won't have results on triad, but certainly more results from the human trials will be forthcoming. But we feel that the dogs with their compressed model of aging will provide a rich data set that you just can't get from humans being treated for 1 year. So where does that bring us? One caveat to all the studies is that timing is going to be everything. There is data from Alzheimer's disease models that if you inhibit mTOR too early, you could actually increase the changes of Alzheimer's disease in mouse models. So timing is going to have to be considered in all of these studies. I think that's sort of a given, but should be underscored in this type of talk. The other thing we're going to see coming up is combinations, okay? So rapamycin plus an IGF inhibitor, rapamycin plus an SGL2 inhibitor. So you're going to increasingly see combinations. And again, as I told you, there's going to be more emphasis on high throughput, individualized medicine, use of worms as a model to reflect your genetic concerns, to see what can be modified for health span and lifespan. So to summarize and leave a few minutes for questions, Geroscience is now over 50 years old and will increasingly impact the way we think about aging, health span, and maybe even lifespan, although most people think we've hit our maximum lifespan. We really need to work on health span. There's plenty of people living to be 110, and many of them are not happy about it. The people, I mean, I have a couple 100-year-olds who are like, why am I still around? Can't you do something about that? And I'm like, it's kind of fun. I do home visits on 3 people over 100, and it's very interesting to hear their perspective. Model systems will range from worms to humans. The Dog Aging Project is providing unprecedented data on a compressed model of aging. And those dogs share many of our environmental features, so we're very interested in looking to see how those interact. And the heterogeneity of the dogs, we think, is a strength of what we're going to find. And just to wrap up the final slide, triad test of rapamycin is an intervention within the DAP and its own clinical trial now. And rapamycin, we think, has a broad basis for intervention, as you can see from the trials we just talked about. We're hoping to improve our recruitment for dogs, but in the triad trial, and we think we will get there, just have to be very creative on creating more sites to make it easy access, because the owners have to come in every 6 months for 3 years. Data on rapamycin is growing, and the future will be built, we think, on repurposed drugs, combination therapies, but timing to minimize off-target effects will be key, as everything else in medicine. And individualized cocktails of anti-aging are here and will continue to develop using AI and high throughput models like C. elegans. And that's it. And I just want to thank all the mentors that got me interested in basic science and biology of aging, many of whom are no longer with us. The DAT founders, Daniel Promislow, Katie Creevy, and Matt Caberlin, the DAT team and the Triad team. And then, of course, Triad is now funded with an R01 from NIA. We're very grateful for that. And there are private donations that we are always willing to accept at the dogaginginstitute.org. Thank you.
Thanks, May. There's a question from Elizabeth Phelan in the chat. She was just hoping that for the benefit of junior faculty, you could describe how you got involved with the Dog Aging Project, given your earlier research career, like how you ended up leading such a big trial